Hi, everyone. Let me begin by thanking Pejun especially for the invitation, as well as Peter and the rest of the CCP-EM organizing committee. This is really an excellent meeting. You guys are very lucky to have this kind of meeting here at this level. It rivals the Gordon Conference and all the other top meetings, but at a scale that's not really matched. So it's really a pleasure to be here. Most of my bread and butter is drug discovery using CryoEM, which is more along the lines of what Sriram talked about this morning. But I want to tell you today about two technology development projects we have to try and help with one of the most common problems in CryoEM, which is that a lot of targets are a bit too small to be studied easily using regular single particle CryoEM. And so the two projects I want to tell you about are summarized here. The first one, I'll be telling you about what we've done to improve Fab fragments as a tool for CryoEM. And the second one is, and that technology is pretty mature now and used at least internally pretty consistently at Genentech, and there's a publication which is under review right now. The second is perhaps a bit more esoteric and adventurous and not working yet. And I'll get to that later. Okay, so the first story then. I think I want to start with a statement that there are a set of targets you might be interested in which are likely too small for CryoEM. How prevalent that problem is, I am not sure, but you can look at the genomes of eukaryotes and prokaryotes and plot the length of the chains that are encoded by genes. And this is something that we took from a review by Todd Yates. And if you take that to be an indicator of the randomly how big a target might be that you select from a genome, then most of the targets from prokaryotes and many of the targets from eukaryotes would be smaller than 50 kilodaltons, which I think you would agree is too small to be easy by CryoEM. Now, of course, many things are oligomeric and in complexes, and so I don't know exactly what the distribution is of targets that eventually we want to be able to tackle. But certainly in our experience, there are targets that are very interesting, which are too small. And that's reflected in, if you look at the molecular weight distribution in depositions to the EMDB. There are very, very few depositions of targets that are under 100 kilodalton, let alone 50 kilodaltons. And that's due to several things. One of them is there's a fundamental limit to the detectability of objects in single particle CryoEM. And the other one is, even if you have picked your particles, the alignability of a very small object tends to be challenging. There's a whole family of solutions to this. One of them is, of course, you get better detectors, better microscopes, better sample prep techniques in general. Another one is you can cheat your way out of the problem by augmenting the size of your target. And so this is one of my favorite approaches out there from the lab of Todd Yates at UCLA, where they design symmetric rigid cages on the surface of which are binding proteins, in this case, DARPins, and you can select DARPins that in theory could bind to any target. And so your target would sit on the periphery of this cage, and it could be very small, but because it's on top of this cage, you can still study it by CryoEM, and because it's a symmetric cage, you get symmetry with it. So this is a really, really a beautiful approach, and they demonstrated recently that they could study famous targets like KRAS, which is about 21 kilodaltons, to a resolution better than 3 angstroms. So great. In fact, this is one example of a class of solutions that we refer to as structure chaperones, things that carry your protein along the process of structure determination and help you do structural biology. And there are a number of examples out there, and I'm just summarizing some of them in this figure, where the maps are colored by local resolution, and the target protein is boxed with the dashed lines. And this is to illustrate that one of the common pitfalls with structural chaperones is that if the chaperone is significantly bigger or more rigid than your protein of interest, single particle image processing can have the alignments driven by your chaperone at the expense of your target protein. And so in local resolution maps, if you have remaining flexibility, or even if you don't have much flexibility, but the target is smaller than the scaffold, then the target protein ends up being resolved at a lower resolution, which is not ideal. We like Fabs a lot. The Fabs are the fragments IgGs and antibodies. They have 2 chains, heavy and the light, and they have a variable domains. And at the top here are the CDR loops, which are hypervariables and determine the specificity of the binding. We use them both for crystallization and for CryoEM. You can, if you have access to collaborators who do immunizations or have libraries and do selections, you can find Fabs against any target in principle. And they're about 50 kilodaltons, 45 to 50 kilodaltons in molecular weight. And so they're a bit small by themselves, but if you have a target that's like 10 kilodaltons, 20 kilodaltons, we think they can work. So why aren't Fabs used? I mean, they are used in CryoEM, but why aren't they ready for prime time? If you download a bunch of human Fab structures from the PDB and you superimpose them on the variable region where the epitope binding happens, you'll see that there is a hinge in the middle of the Fab, which is very flexible. You can measure this as an elbow angle. And so in human Fabs in the PDB, typically you'll have an elbow range of about 10 degrees, which ends up being significant. When we do CryoEM using Fabs on top of our targets, like we did here with CD20, which is a membrane protein, we end up with class averages where the constant part of the Fab, constant domain part of the Fab, is kind of smeared out because of this flexibility from particle to particle. And so, in renderings with an isocontour level, you get sort of, you lose the constant part of the Fab. So this flexibility, we think, is a significant problem. And Jennifer, who was a postdoc in Sudhamsu's lab in the structural biology department a few years ago, starting with the observation that rabbit Fabs have much less variability in their elbow angles in the structures, decided to see if they could come up with designs to rigidify this, to lock this elbow in place so that the Fab would become a rigid 50 kilodalton scaffold for structure determination. And so over a period of a few years and over dozens and dozens of constructs and designs, Jennifer expressed a bunch of Fabs against targets that we already had in-house, made sure that the mutations, selected mutations that didn't interfere with the binding affinity. And then we didn't know a better way to do it. We did a lot of CryoEM to evaluate the flexibility of the Fabs and the mutated Fabs. And this was an iterative process. When there was an initial success, then we iterated with more mutations on top of the existing mutations. So we needed a test bed for this. And even though eventually this is meant for small targets, we started with a larger protein that we were doing in-house anyway. It's an extracellular tetramer, and it's each monomer of the tetramer can be bound by one Fab the wild type Fab that we started from, and we already had a structure of this. So the total complex size was 320 kilodaltons. It meant we could quickly iterate over the mutants that we were testing. And so, before I go any further, all the mutants that I'm going to show you are the ones that had preserved affinities. So none of the mutations we did affected the binding to the target. This is shown in this table here. So this is a fairly easy complex to work with. The orientation distribution isn't ideal, but you get decent maps out of it. And our initial wild type reconstruction was at about 3 angstroms, 2.9. This is colored by local resolution, estimated using the method implemented, for example, in Reliant with a windowed local FSC. And you can see, as expected, that the flexibility at the hinge of the Fabs means that the constant domain is less well resolved. The first mutant was really inspired by these rabbit Fabs. They turn out to have a disulfide around the hinge that kind of holds it in place, more or less, and seemed to give marginal but noticeable improvements in the local resolution, both of the target itself in the center here and as well as in the Fabs. Perhaps a slight improvement in the constant domain, but that wasn't so clear. But then, as I said, Jennifer iterated through dozens of constructs, eventually found a way to add more mutations on top of the initial ones. And from generation to generation of our mutants, we ended up getting better and better resolutions, eventually getting to better than 2.5 angstroms, and with somewhat better resolved constant domains, but more importantly, the core of the target was better resolved. There was something else I wanted to say about that, but I'm forgetting. So More important than resolution numbers, here are some examples of details of the maps, and you see by the time we were done and had added lots of what we think are rigidifying mutations, we were seeing really more details in the reconstructions. And here is another example. I showed you earlier a publication we had with CD20 and Fabs, and the constant domains were completely smeared out. When we used the most rigidified mutants that we've come up with so far on CD20, we went from this situation in wild type. Well, I now remember what I want to say about the previous slide. We tried to control it as closely as we could. So this was same microscope, same detector, same number of images, but different grids. So we couldn't, we can't guarantee that the ice thickness and everything was exactly the same, but it's a pretty well-controlled experiment. So going back now to where I was, wild type looks like this, rigidified looks like this. This is an example where really you can see very clearly that the constant domain of the Fab is really better resolved in the rigidified version. And I think the reason we weren't seeing it before is because we had this really large target with 4 Fabs on decorating it, and the alignments were really driven. The center of gravity, if you will, of the alignments was the core of the target, whereas here, CD20 is a pretty small protein with, only on the order of like, say, 40 kilodaltons of ordered mass in a micelle. And so here the Fabs, they can drive the alignment a bit more, and you really fully see the benefits of the rigidification. But actually, this is slightly cheating, this figure, because what actually happened is this. In the initial refinement of the rigidified Fabs, it was this map. And you see actually that the CD20, the target itself, was kind of smeared out, more so than in the wild type. So what I just said about the rigid Fabs driving the alignments was actually at the expense of the target. But what you can do in these cases is do local refinements. And so by doing a local refinement just on the membrane protein and the variable domains, this situation was improved. If you make the mask even tighter just around the membrane domain of the protein, you get better resolution of the transmembrane helices. Admittedly, this is still not perfect. This is a pretty flexible protein. And then if you combine all of these, there are programs to do this, like in Phoenix, combined focus maps. You take the best bits of every focus refinement and you combine them into a single map. You get something where you get good resolution all the way from the constant domains to the end of the membrane protein, but it's a composite map. So now I've touched on this a couple of times. You see that when you have a structure chaperone that's very rigid, if the target itself is very large, they can fight against each other in the alignments. If the target is small but flexible, the Fab wins the alignment battle, and then so you have to be a bit selective about how you design your structural chaperones in a way, depending on the target. Our use case, though, was really meant to be small proteins. So do these rigidified Fabs help for small proteins? And I'll show you 2 examples where we're pretty convinced that they did, and we have more in-house that we're not yet disclosing. So this is a 26 kilodalton protein, and it purifies pretty well. If you do an SEC with a slight excess of the target proteins, this is what the profile looks like. It's a clean complex. Put it on a grid. The class averages look something like this. Pretty straightforward image processing. We didn't really have to play any tricks here, even though this is a small target. And this is the final map. So overall, including the Fab, this is a 74 kilodalton object, and the target is up there in pink. And in this case, there seems to be a nice match between the size of the structure chaperone and the target. And so the alignment is really pretty uniformly good over the full reconstruction. Still, some flexibility in the hinges, and the constant domain of the Fab is slightly well resolved, but really that map is a pretty high quality 2.7-ish angstroms. And here is just a slab through the complex. Again, the Fab is on the right in the yellow and white, and then the target is in pink. So lots of waters and features that you would expect at this resolution. And these are some little details of the maps, of the map. Okay, a second example we have, this is not yet published, but it's under peer review right now. So the proteins are anonymized, but I can tell you this is one of the ones that Sriram was talking about. lost my pointer. Yeah, the small protein, 21 kilodaltons. So anyway, the complex is also pretty well behaved. Again, on the grid, you know, the image processing was pretty straightforward. Not a lot of effort needed to go there. Here we got to about 2.8, 2.9 initially, and then a local refinement to just better than 2.8 maybe. And this is an isosurface rendering. This protein, 21 kilodaltons, also happens to have two small molecule ligands bound to it, which were well resolved. This is the local resolution map of the global refinement. And then this is with local refinement over just the variable domain of the Fab plus the target. But either map is pretty good. These are the sorts of details that you can resolve in these maps. So these two examples are meant to be illustrative of what you'll have to take my word for is a larger set of structures we've enabled in-house with some very small targets. These are pretty representative. There are, of course, some caveats with this technology. You need to have access to a collaborator or someone who can provide you with monoclonals. You have to select the ones that bind with high affinity. Not always is something that's high affinity by SPR will that Fab survive CryoEM when you freeze grids? There are things that happen sometimes where occasionally you'll get dissociation for reasons we don't understand. This is not like the silver bullet, but we think if you have a small target that is really valuable to you and you're going to study it over time, it may be worth investing in a sort of antibody discovery or panning campaign to get some binders. And then you can rigidify them. We're going to publish exactly the recipe for which mutations you should put in. And then you can try it with CryoEM rather than crystallography. So that was story one. And that works. The next, everything else I'm going to say, I would say don't try at home. It doesn't really work yet. But I just want to give you a flavor for why we were excited about this and where we are in the process. Okay, and it has to do with soft landing. So a few years ago, my colleague Liz Hecht, who's a mass spectrometer at Genentech, came to me and basically said, what if I could fly for you individual proteins, each suspended in a tiny nano droplet of solution, keep it hydrated all the way, and then land it softly on a grid that you would tell me how to keep cool and transfer to the microscope. And then you could look at it. And I thought that was very interesting. Kind of science fiction, but you know, she was proposing to do the hard work, so I was like, okay, let's try this. Now, soft landing is not new, and I'll show you in the next slide some previous examples. People do it for different motivations. Some people do it because they'd like to have like a mass spec-like instrument that would select different masses and land them on the grids and purification, basically. Other people do it for different reasons. The thing that I was most interested in is there is a fundamental limit in CryoEM to detectability, right? Depending on exactly which version of Richard Henderson's papers you read, it's something like 40 kilodaltons, or Chris has new estimates, you know, depends on the background and stuff. In single particle of purified proteins, the main problem really that we don't think about, we take it for granted, is contrast matching. The water molecules and the other buffer molecules in our ice layer almost contrast match our protein, which means we have pretty terrible contrast, and the detectability is impaired by this. But if what Liz was proposing could be made to work, ideally we would have individual proteins surrounded by a very thin, minimally thin, I don't know exactly what that means, hydration layer, so it would still be a hydrated native state, but without the ice layer that we normally have, and so the contrast should be amazing. That was what I was thinking. And so I thought maybe we could then eventually take this to smaller proteins and that we could align in a way that we can't currently. Now, there are probably a million red flags that have gone up in your minds as I was saying all this, you know, and rightly so. Air-water interfaces are a big problem. This is going to be taking a protein and exposing it to the air on all different sides all the time. Terrible idea. There could be denaturation when it lands on the surface. And then one thing I'm particularly worried about is that we don't really know, maybe you do, but I don't, how how CryoEM protects from radiation damage. But one of the dominant theories is that the physical encasement of our proteins in a solid state by the vitreous ice means that even as radiolysis happens with the first inelastic event, the protein cannot fall apart the way it might want to otherwise, because it's just constrained in space. But now I'm proposing to remove that layer, which could lead to terrible consequences with the first damage event. So anyway, lots of reasons not to do this, but we thought it would be a fun experiment or set of experiments to try. Okay, so we went ahead and started a project. Now, there's lots of different approaches that people have tried with soft landing. The one that I had been aware of previously was actually a collaboration between Helen Sable and Carol Robinson, which had been published some years ago, where they had basically modified a mass spec instrument to have a stage for room temperature continuous carbon grids. And they then negatively stained them and looked and showed that CryoEM was actually preserved, at least it was still in the multimeric state that you would expect and so on. It never got to high resolution though, so there were already hints back then that this wasn't really a perfectly happy CryoEM, and I'll show you more about that in a minute. Broadly speaking, two different categories of systems have been proposed. Some of them are true mass specs with an EM grid at the end under vacuum. We went with an approach where we keep everything under atmosphere at all times so that we could control, hopefully, humidity and so on to avoid complete evaporation. Those of you who are not familiar with that field, the reason it's called native mass spec is largely because of experiments like this, where if you fly an enzyme through a mass spec, you land it on the surface and then you rehydrate it, many enzymes preserve activity. And so this is one of the arguments for calling it native. Our system starts with electrospray, like in most mass specs instruments. We are concerned, though, that during one of the things that happens in electrospray is droplets fission and break apart, and they very, very quickly shrink to more or less the size of the target proteins. We would like to slow that down so that each protein is surrounded by a generous layer of water, at least at the beginning. And so we have a trick where we use an alpha particle emitter that ionizes the air, and all these charges in the air sort of neutralize the charges on the droplets, which slows down this process. And we hope that that helps keep the proteins more hydrated. So anyway, by the end of this, we have a set of droplets where we've either neutralized them or left only one charge. It's like a Poisson distribution of a number of charges. And later, we want to select only the +1 exactly nanodroplets that contain exactly one protein. And how do we do this? We use a device called a differential mobility analyzer, which is used in environmental sciences and so on to characterize particles. And the way this works is, again, this is under normal atmospheric pressure. Think of it as a tube, and there's like a gas flow through it that carries your droplets that you want to analyze it. And down the center of that tube is an electrode. You apply a voltage to that electrode, which deflects ions according to, well, how charged they are, but also their collisional cross-section, so how big they are. And you can tune the voltage so that you select for a particular species of ions. And so our idea here is to select for exactly +1 charged ions that contain exactly one protein, so the diameter of a single protein complex. And then once that's done, we go on to a landing chamber where the grid is kept cold. The results I'm going to show you were obtained at temperatures between -20 and -50 Celsius. So not liquid nitrogen cold, because if we did that, then we would get lots of condensation from the atmosphere, no matter how dry we try and keep it. But that holds a pre-clipped grid, and it has a continuous either graphene or carbon so that the proteins don't fall free. Okay, so that's the basic idea. I'll skip over this, but this is quite different from the other talks you may have seen recently about soft landing, because we're at atmosphere, and we have singly charged ions with much lower energies. This is the first time that we had a pretty successful experiment in the sense that we could recognize what we had landed. And this was CryoEM, landed on a grid that was kept at about, we think, -20 Celsius. There's a continuous graphene support film, which unfortunately wasn't very clean at the time. Later, I'll show you much cleaner backgrounds. But each of these high contrast objects, we believe is CryoEM. The stuff in the background, the smaller objects, we believe is mostly contamination from the film, but I'll show you cleaner results later. On the right is from the same prep of CryoEM, which wasn't particularly nice, is a just conventional Vitrobot prepared grid to give you a visual frame of reference. But thankfully, Helen showed lots of CryoEM yesterday, so you should have it in your mind's eye already. You know, on the top view, it's a circular projection. On the side, it's a rectangular projection. You know, it actually looks pretty sharp and, you know, really geometric in regular CryoEM. And what you can already see, I think, here is that the rectangle is pretty rounded and doesn't look quite right. But very, very high contrast. I hope you'll agree, and I'll show you more example of this. I should say this is pretty defocused, so you get these sort of fringe-like features around the particles. We next moved. I want to show you a few slides of AAV capsids, which are nice test objects for us because a big deal for our method is we want to keep the particles hydrated, and AAV capsids are hollow objects. And so if you keep them hydrated, you would expect that they would be much darker than the background. And this was the first time that we saw AAV capsids successfully, but it wasn't really completely successful because, as you can see here, the pixels on the inside of the capsid are much brighter, which suggests that these things were actually empty. Somehow the buffer had leaked out of these capsids before they landed. Not only that, but so this is an example much closer to focus, clean background, I hope you agree. But not only that, so this is an example where we've solved that problem and we'd landed AAVs that were still carrying buffers within them. But some of them were severely damaged. So the capsid looked kind of crumpled in some way. Here's another example, slightly more defocused, so you start to see the ringing. And I think you can appreciate, you know, this is not what AAV is supposed to look like. So here are some class averages. You know, we can collect data sets from these. 3 different experiments at the top. For reasons we don't quite understand, this was more dehydrated. The capsids were depleted of their buffer. At the bottom, they were more full. This is interesting because here you have a good example of a full capsid, as in full of buffer, and surrounded by a slightly larger than itself droplet. And then the edge of the droplet is what gives a lot of contrast, and you get these ringing artifacts from the defocus. CTF models that we use for normal single particle processing assume very low amplitude contrast, which isn't the case here. So it's difficult to get really nice CTF correction with these images. But you know, long story short, we can't average these to anything coherent. We think that there's some kind of denaturation that's happening, even though we have kept them hydrated in some respects. This is going back to CryoEM, closer to focus again, pretty clean backgrounds. You can sort of see different views. But again, when we average them, it really doesn't work the way it should. This is amazing contrast microscopy. Really, in an individual image, you should see all the features that Helen was showing yesterday. You know, very clear double barrel. On the top views, you should see 7-fold symmetry. The best we can do at the moment is if we average a much larger number of particles than we should need to, we can sort of see 7-fold symmetry near the core, but not through the structure. And you know, I've shown you AAV capsids, but CryoEM is kind of like AAV capsid. Helen was showing sections through it yesterday. It's full of enormous cavities that are water-filled and later by substrate. And I think what we interpret this to mean is that despite our efforts so far, we haven't controlled evaporations tightly enough from these droplets, and that by the time they land, at some point in the process, they have been denatured, we suspect, by evaporation. But the only way to test that really is to go back and redesign every part of the instrument and figure out where we went wrong with our humidity control, basically. So this is work that is now ongoing with Henry Benner and his company in California called IonDX. He's really the expert in this ion mobility field, and in a collaboration with Genentech. So that's the 2 stories. Thank you very much for your attention.